Okay, thank you. So we can start the presentations now. Our first presentation is by Mary and Sai from Baylor University. You may share your screen and begin when you're ready.
All right, can everyone see our screen?
Yes, I can.
Awesome. Good morning, everyone. My name is Sai Sagireddy.
And I'm Mary Mercero.
And today we're here presenting the discovery and analysis of Bacteriophage casserole. We're two freshmen at Baylor University.
So we did this through the C-Phages program, which is a one-year research-based course for freshmen. The purpose of C-Phages is to discover and analyze bacteriophages. And this is important work because bacteriophage research has led to various research advances, most famously in fighting antibiotic resistance, but also in agriculture, the food industry, and many other fields. So through this program, we discovered Phage casserole, which has many unique and interesting characteristics.
The C-Phages program is divided into two parts, discovery and analysis. I'll first walk you through the first part of discovery. It consists of four main steps: soil collection, soil washing, purification, and amplification. So phage casserole was found under a bald cypress tree near the Waco Creek near the Baylor Sciences Building. It was isolated from a mildly acidic soil sample, and after we conducted various tests, we determined that the different proportions of water, clay, sand, and silt allowed us to confirm that it was from sandy clay soil. Figure 1.3 shows the exact coordinates and the location where we found bacteriophage casserole. After we moved forward from soil collection, we moved on to soil washing. So the soil was washed with PY broth, and then it was placed into a vacuum filter, as you can see in figure 1.5, and filtered so that we could obtain a lysate from that. And this lysate was then enriched with Arthrobacter. Arthrobacter is a soil organism. It's a bacteria, and it's grown on pica media at 22 to 28 degrees in the lab, and it rapidly reproduces every 2 to 3 hours. So that's one of the reasons why Arthrobacter is used, because it's widely available. And bacteriophages, the main goal of it is to kill bacteria. So as you can see, we then placed our lysate and with top auger and Arthrobacter and performed plaque assays and spot tests. Figure 1.4 shows that there is a clear spot on 10 to the minus 1, which confirms the presence for bacteriophage. Then we moved on to purification. Purification tells us that we only have one specific phage present. This is very important because we wouldn't want to have multiple phages present, which would disrupt the entire experiment. Casserole underwent 3 rounds of purification, and we had multiple plaque sizes. One being a larger 1 milliliter and a smaller one being less than 1 milliliter. The plaques were clear, showing us that casserole was part of the lytic cycle. So bacteriophages can be part of 2 specific cycles, the lytic and the lysogenic. And 80% of phages undergo the lytic cycle and 20% undergo the lytic cycle, under the lysogenic cycle, sorry. And the lytic cycle, if you get clear, transparent plaques, it represents that the phage is part of the lytic cycle, which is in the case of phage casserole. If it's the lysogenic cycle, we will see cloudy plaques.
All right, so as I said, after purification, we knew that we had isolated one clonal population of phage, and this allowed us to move on from discovery into the analysis portion. So the analysis portion involves DNA extraction, electron microscopy, and bioinformatics. So to do the analysis portion, we had to amplify our lysate to a higher concentration of phage. We call this a titer. And so once we got our sample up to a high enough titer, we were able to extract DNA. After we extracted DNA, we ran it on gel electrophoresis in order to find out, kind of predict what cluster our phage is a member of. Phage clusters are important because phages in the same cluster share a certain amount of DNA sequence in common with phages in that cluster. So as you can see on our gel electrophoresis on well 5, we got pretty clear results for the AV cluster. But in well 7, we also got a little bit of band showing up on the AP cluster, but we thought it was probably AV due to the heavy results. And if it wasn't the AV cluster, this would be very exciting because AV cluster is a very rare phage to find. After electrophoresis, we moved on to TEM, transmission electron microscopy. And this allows us to magnify the image of our phage 50,000 times so that we can see it. And Sai and I were very excited to perform TEM because we had been working with our lysate all semester, and then we finally got to see what this phage looked like. And as you can see, phages have 3 different morphotypes. The type of phage that you probably have in your mind would be Myoviridae, which kind of has a medium tail and pretty prominent tail spikes. There's also Syphoviridae, which has a long tail and short tail spikes. This is also a pretty common phage to find. And then there's also Podoviridae, which has a short capsid head and a short tail. And this is a very rare type of phage to find. And as you can see, in that TEM image, you see a protein capsid head and a very short stub tail. So this showed that casserole is a Podoviridae, which no one at Baylor through the C-Phages program had ever found a Podoviridae, and it's just a very rare phage to find in general. And then we noticed that on the Phages DB database, which is where everyone records information about their phages, that in cluster AV, all the phages were Podoviridae. And so this kind of further confirmed our predictions that phage casserole is in the AV clusters. This was all very exciting for us.
So TEM was performed in December of the first semester, and throughout winter break, we sent all our samples for genome sequencing to the University of Pittsburgh, and our results were very, very exciting. The results confirmed that phage casserole was the first ever AV cluster found at Baylor University and the 6th one ever discovered through this program. And after we got the results this semester, we moved on to annotation, where we explore the genome of phage casserole. Figure 1.11 shows our bioinformatics software, and the green part of it shows our forward genes, and the red shows our reverse genes. And phage casserole has 58 confirmed genes, which we are exploring this semester. Figure 1.12 shows us the Famirator map. Famirator is a program that allows us to explore all the genes of all the different AV clusters. This is important because the significance of a cluster is that each phage within that cluster has 50% similar genes. So once we have verified that phage casserole has 50% or more of these genes, that means that it is definitely part of the AV cluster and allows us to confirm that the genes present there do exist.
All right, so as we were exploring these genes through gene annotation, we noticed that casserole has a lot of kind of weird, honestly, information in its genome. Casserole has overlapping forward and reverse genes, which is very unusual. It's kind of hard to imagine how this works during the transcription process. We did research through the C-Phages forum to see if anyone else has come in contact with this, and we didn't find a lot of information about it. So we're looking forward to more researchers coming in contact with this and learning how this works. We also saw that although casserole, we isolated with Arthropacter, we noticed that some of its genes share sequences with bacteria other than Arthropacter. And this made us wonder, did we isolate a bacteriophage with something that wasn't its host, or did it pick up these genes from elsewhere in the environment? So that's also pretty weird. We also saw that 7 of its genes are orphans. And so what that means is that these genes are not found in any other phages. They're unique to casserole. And finally, we saw that genes 2 and 3 are duplicates of genes 58 and 59. So 2 genes in the beginning and 2 genes in the end directly match each other and are also orphans. So overall, casserole just has a very diverse genome and a lot of information that we're looking forward to exploring further.
Phage casserole is such a unique phage with so many different unique characteristics, and it is nothing like the other phages that have been found at Baylor University. We are very, very excited to explore this further this semester and next semester. Some of the things that we plan to do is that understand why genes 2 and 3 and 58 and 59 are so similar to each other. This is a characteristic that we have not seen in any other genome within a phage. And we are wondering if casserole is really an Arthropacter phage, because when we blasted this on NCBI, it had similar sequences to other bacteria, as Mary said. And this is very, very interesting because it's not something that we saw in common with other bacteriophages discovered. It has overlapping forward and reverse genes, and this is also something we have not seen in other phages, and we do not know the significance of this yet. But as technology advances and more AB clusters are found, we are really, really excited to compare and contrast and see and understand what all of this means. Thank you so much for your time today, everyone.
Any questions?
I
Okay.
guess I'll go. Well, go ahead.
Oh, thank you. I have one. So did you just did the sequencing to the DNA you extracted from the bacteriophages?
Yeah, so once we extract that DNA sample, we send it to University of Pittsburgh, who is also involved in the Phages program, and they sequence it for us and then send it back. And then we're able to analyze it through DNA Master and other bioinformatics programs.
Sounds good. So have you ever tried to look at the, I'm not sure if it's necessary because my research is related to RNA sequencing sometimes. So have you ever tried to look at the transcriptome of the bacteriophages?
Yes. Do you want to answer that question?
Sure. So I think that's one of the goals of the FAMRater program that we use. It allows us to compare the specific genomes of different phages, and that is a part. So FAMRater is an open source platform that researchers are continuing to develop as time goes on. And I think that is one of the things that they are planning on developing there. So we're excited to see how that goes in the future.
Mm-hmm. And also, like, as we're looking at the genes, we're also able to look at the protein sequences. And so we also compare like the protein structures that each gene creates through like HH-pred. We're able to look at like the different structures that the amino acids probably make. And that also allows us to like compare genes from other phages.
And phage casserole has overlapping forward and reverse genes. And when we look at that, we sometimes wonder where the RNA polymerase will exactly bind on that genome. And if we do conduct RNA sequencing at some point in the future, that will really help us understand what that overlapping forward and reverse genes will do.
Sounds good. Thank you.
So I know that they've sampled a lot outside on that creek area. What may have made that a unique sample compared to the probably hundreds of samples that were taken out there?
So I think something that has been so unique to our C-Phages class this semester is because of COVID, we took all our soil samples for the class from one specific area. So those were 10 soil samples that were taken from this same area. And our class, out of the 10 groups, 2 groups tested positive for phage. The other one was phage dynamite, which is part of another common cluster. And this one is phage casserole, which is part of the AB cluster. And that was the only difference that we were able to see, that we sampled at such close proximities. And it was so interesting to see that 2 completely different phages existed at the same space. We have all of these soil samples stored in our lab with Dr. Tamara Adair, and we hope to some point in the future explore this in the wet lab to test the different soil samples, understand the characteristics, if there's a difference in the type of soil, or see what is the exact difference in these soil samples that allowed it to host different bacteriophages.
Yeah, it was pretty interesting to me because even though we took all the soil samples from the same area, they were different types of soil, like whether it's sandy clay or, so the components were different. So we're looking to like analyze those soil samples further and see like, is there different phages that bind a bacteria better in different soils? So.
Yeah, that area also went through a flooding. I can't remember. It wasn't obviously last summer, but it might have been the summer before. I can't remember because of all the COVID stuff. My memory's wiped clean. So it depends on where you took your sample. If it was close enough to the water's edge, that water's edge went up above the tree trunk area. So you may have some, maybe some environmental factors that has occurred prior to, your sampling.
That's so interesting. And I will say that the other phage that the people in our lab isolated was shared all but 2 base pairs with a phage found by our TA 2 years ago. And so it really seems like it was the exact same phage. So not only did we find a completely unique phage, but the other phage that was found has basically already been discovered. So it's just very interesting how all that is existing in the same soil.
Yeah, it'll be interesting too after this freeze that we had, how that's going to change all the dynamics. You know, when you go below zero for as long as we went, that's going to change what's going to appear in the environment over the next year.
Very true. That's interesting. Any other questions?
I think that's all the time we have. But thank you, Mary and Sai, for a wonderful presentation. Our next presentation is by Ashley from Tarleton. Before we begin, though, judges, do you need more time to finish writing any kind of feedback?
I'll have to finish mine after we finish all the presentations, but I've got enough notes.
Okay.
I'm doing well.
Okay. Sweet. Then we can start the second presentation. Ashley, you may share your screen and begin when you are ready.
Good morning, everybody. Can you all see my screen?
Yes, it can.
Wonderful. Well, my name is Ashley Suris. I am a senior at Tarleton State University. And today I'll be presenting my research on the isolation and annotation of cluster EB bacteriophage Indilu. Bacteriophages or phages are viruses that selectively infect bacteria. Concurrently discovered in the early 1900s, phages were studied for their ability to kill bacteria and explored as an antibacterial therapy. Despite being estimated as the most numerous organisms on the planet, research into phages received minimal interest in Western medicine after the development of the modern-day antibiotic. Combating the growing issue of antibiotic-resistant infections has renewed scientific interest in the application of phage therapy as a potential alternative treatment. In conjunction with the C phages and C genes program from the Howard Hughes Medical Institute, students are isolating bacteriophages from the environment in an effort to build a library of novel phage genomes, identify lytic and defensive genes, and expand existing databases for use in future phage applications. Bacteriophage Indilu was directly isolated from a soil sample found near a horse barn in Stephenville, Texas. The sample was suspended in pica media and incubated and filtered with a 22 micrometer filter. The sample was plated with host Microbacterium foliorum on pica plates with a soft agar overlay. Following 24 hours of incubation, small lytic plaques formed on the plate. A serial dilution and plaque assay was created by picking one of those plaques, and a second serial dilution was conducted to analyze plaque morphology and verify consistency. Webbed plates from this plaque assay were flooded with phage buffer to collect low volume lysates, and a titer was calculated. We used webbed plates for this purpose to maximize the concentration of phage in the lysates. Unwebbed plates have too few plaque forming units to yield sufficient concentration, but cleared plates in which the phage has already lysed all of the bacteria are also excluded because phages are actually quite unstable outside of a host. Using that titer, we calculated the number of plaque forming units needed to create additional webbed plates. The corresponding volume of low volume lysate was used to create 8 additional webbed plates. These resulting plates were flooded with phage buffer for high volume lysate collection, and high volume lysates were generated as compared to just moving forward with low volume lysates to promote product stability for storage and to increase our chances of success with sequencing and visualization. These high volume lysates were serially diluted and plated to calculate titer, and the resulting plates are shown here in image 1. We used a series of additions, incubations, centrifugations, and supernatant removals to extract purified phage DNA from these high volume lysates. We started with nuclease to degrade any remaining bacterial DNA or RNA that was present. Zinc chloride precipitated the phage, and then we pelletized that. The pellet was resuspended in TES buffer, and then after resuspension, we added proteinase K to eliminate any remaining nuclease activity. After that, potassium acetate degraded phage capsids, and EDTA within the TES buffer protected the newly exposed phage DNA. An ethanol wash and resuspension in nuclease-free water yielded purified phage DNA. DNA concentration and purity was verified with a nanodrop spectrometer and qubit fluorometer, and a sample was sent out to the Pittsburgh Bacteriophage Institute for next generation Illumina sequencing. Gel electrophoresis of the DNA was run in 0.8% agarose gel with lambda DNA Hindi 3 digests and a lineup of restriction enzymes to visualize the genetic fingerprint. Only a single band was present after enzyme digestion. We think this could be either due to possibly chemical protection like methylation or simply an absence of cut sites. The high volume lysates we collected were attached to a 300 copper mesh grid and negatively stained with 1% uranyl acetate for transmission electron microscopy visualization, which revealed Cyphovarity morphology, a capsid head diameter of approximately 60 nanometers, and a tail length of approximately 140 nanometers. Bioinformatics software was used to conduct comparative analyses to assign putative functions to protein coding regions and verify start site and coding potential of these genes. Aragorn identified a TNA responsible for recruiting glycine during the replication process, and Famerator generated a gene map that was used for comparison to the protein coding genes of other bacteriophage genomes. Of particular interest was structural similarities predicted by HH-pred between gene 70 and CRISPR-Cas9 endonuclease proteins due to the antiviral nature of Cas9. Primarily observed in bacteria and only recently discovered in megaphages, CRISPR genes, including Cas9, serve as bacterial defense against phage infection through the incorporation of short genome sequences used in the future for targeted viral destruction. The presence of a structurally similar gene within a moderately sized phage prompted further exploration, which revealed homology specifically within the H&H subunit of Cas9, which is directly responsible for the sussorial effects on targeted DNA. Aligning these proteins in MegaX revealed several conserved residues and a DHXXP motif common among many Cas9 orthologs. Additionally worth noting was the presence of the highly conserved and catalytic histidine residue and downstream asparagine residue. Sorry, it was all y'all's faces were blocking that residue. I couldn't see it. As is also common in Cas9 orthologs, the second anticipated histidine residue of H&H endonucleases was replaced instead with a second N residue, resulting in an HNN protein. In silico modeling software revealed the beta-beta-alpha subunit expected in H&H endonucleases as present, although slightly modified with short alpha helices preceding the second beta sheet. This modification was also observed in an E. coli phage in a paper by Whitich et al., which we'll discuss here in a little bit. We conducted evolutionary analyses in MegaX using the p-value method and bootstrap test. This revealed that the nearest evolutionary neighbor that we tested to Indilu 70 was a Cas9 protein found in A. nislundii, which makes sense because we're testing Actinomyces bacteria. Phages DB identified gene 70 as belonging to FAM 54465. Review of this FAM revealed 19 clusters in which this gene was present. We generated an upset plot to compare gene location, phage life cycle, host, and whether all genes within that cluster contain the gene. In all but one cluster, the gene appears at the extreme ends of the genome and is present more often in temperate phages than it is in lytic phages. The most common overlap was in clusters of temperate phages hosted by Gordonia, in which the gene was located at the C terminus, and not every member of the FAM carried the gene. Cluster EB was the only cluster carrying this gene hosted by Microbacterium, and one of only 3 clusters which were lytic. We generated a dot plot to compare Indilu with 2 additional cluster EB bacteriophages and 3 non-EB bacteriophages that contain the gene. The alignment outside of cluster EB was very obvious in both a 10-word length test and a 5-word length comparison. And you can start to see in the 5-word length comparison the similarities with the non-EB phages. The alignment was primarily seen at the beginning of the genome, which we also verified in a spot test with MegaX alignments. A publication from Kala et al. Provided strong synthetic support for clarified endonuclease function of gene 70. This paper showed that in the previously mentioned E. coli phage, HK97, there was a protein called GP74 at the C terminus of the genome, which interacted with the large and small terminase subunits at the opposite end of the genome. And it served to cleave concatenameric genomes into single genomes during rolling circle replication within the host. GP74 also served crucial roles in phage head morphogenesis. When it was cleaved, phages formed functional tails separate from complete heads. IndyLu shows synteny with the location of GP74 very near the C terminus of the genome, and IndyLu's terminase subunits are located at the extreme N terminus of the genome. This syntenic pattern was also observed in a sampling of additional clusters carrying this gene. Alignment in mega-X of HK97 GP74 and IndyLu70 showed 15 conserved residues. The alpha helix present between the beta sheets and IndyLu, which is absent from many H&H endonucleases, was also present in HK97 GP74. In conclusion, bacteriophage IndyLu is a lytic phage with a 41,958 base pair genome, 3' sticky overhang ends, and is 66.2% GC content. IndyLu carries a tRNA responsible for recruiting glycine during the replication process, and Annotation identified 71 protein coding genes, including one gene with structural similarity to the H&H subunit of CRISPR-Cas9. Further directions include submission to GenBank, verification of function of IndyLu gene 70 in phage replication, and exploration of the evolutionary benefit to both phage and potentially host of a bacteriophage carrying a Cas9 H&H endonuclease ortholog. Thank you all very much for your time, and I'm happy to answer any questions you may have.
Well, I'll go first then. So I was noticing that you know, you got your sample from like a horse area or something like that. Have y'all been taking samples from that area in the past with your program?
You know, I believe they have. It was actually a classmate of mine that collected the sample, but I know it's out at our agricultural center, and I believe several other students have tried to get samples from that area.
So are y'all GPSing these sites too?
We are, I don't know how like specifically GPS it was, like we didn't use special tools or anything, but we do document where it came from and pull that up with Google Maps to get the latitude and longitudinal coordinates.
Okay.
Okay, I just have a quick question. So about the phage isolation, I'm really curious, how did you isolate the phages from the bacteria?
So we did that with the serial dilutions and the plaque assays. We collected those lysates, and then the serial dilutions and, excuse me, not the serial dilutions, the lysates collected were plated. I am butchering this answer, I'm very sorry. Could you ask the question one more time?
Yeah, I'm just curious about how did you isolate the bacteriophages from the bacteria from the soil samples?
Okay, so after we filtered it with a 22 micrometer filter, we were able to plate it to collect those lysates. Mm-hmm. And after 2 rounds of purification with plating, we could separate the phages out with the nuclease that we added. That after we had the high volume lysates, the nuclease degraded any remaining bacterial DNA and RNA.
Ah, I see. Thank you.
Thank you.
Thank you, Ashley, for a wonderful presentation. Our next presentation will be by Gustavo from Tarleton as well. And again, before we start, judges, do you need any more time?
Good.
Good.
Okay, then Gustavo, you can share your screen and begin when you're ready.
Thank you so much for that. Let me just convert it to, okay. Hello, my name is Gustavo Vasquez, and I'm a biomedical science major. Hold on. Okay. I'm gonna restart that. Hello, my name is Gustavo Vasquez, and I'm a biomedical science major from Tarleton State University. Today, I want to present to you all about the discovery of bacteriophage that was isolated, amplified, completed DNA extraction, and characterized, which was followed by annotation using alumni next generation sequencing. This virus was named Fizzles by Skylar Adams and Gabriel Spotts. Fizzles is also considered cluster EG bacteriophage. Before moving forward with the presentation, I want to talk about what bacteriophages are and how they are a potential weapon against bacterial infections. Over the last few decades, bacteria are increasing antibiotic resistance through formation of resistant mechanism by using instructions provided by their DNA. Often, resistant genes are found within plasmids, which are small pieces of DNA that carry genetic instructions from one germ to another. This means that some bacteria can share the DNA and make other germs more resistant. Some common bacteria that are antibiotic resistant and are becoming a threat to us are MRSA, vancomycin-resistant Enterococcus, and among others. Bacteriophages are able to infect and kill these bacteria and have the potential to become the next generation treatment for antibiotics. They are also the only therapeutic agent that regulates itself at the sites of infection in the case where the infection has been killed. So overall, our goal was to isolate, name, sequence, and analyze fizzles. The isolation of fizzles began by collecting a soil sample from an ant nest in Stimulio, Texas. This was followed by direct isolation using liquid media to extract the phage from microbes and particulate matter in the soil sample and filtering it through a 0.2 microliter syringe filter. Using host bacteria, Mycobacterium flororum, a plaque assay was performed to visually confirm the presence of a phage particle in the sample. This was done by mixing the host bacterium with the liquid sample and growing on agar plates for 24 hours. The plaques assay shows small turbic lytic plaques. These plaques illustrate that phage fizzles was able to infect and replicate within the bacterium host as well as lysing it through the process. So to retrieve phage fizzles, a plaque was picked using a stereotip to gently stab the top agar in the center of the plaque. The tip was then placed into a phage buffer in a microcentrifuge tube, and the solution was further mixed well by vorticing it for 5 seconds. Further experiments included serial dilutions to manipulate the number of phages in the sample. In this protocol, we used tenfold dilutions, meaning that the concentration of the phage in each tube was 10 times less than the previous tube. So 10 microliters of undiluted phage sample was added to 10 to the 1 tube and vorticed for 5 seconds. This was done all through 8 tubes. Next, serial dilutions had to be plated according to the Plaque Assay protocol as mentioned above. Non-agar plates were obtained and labeled with reference to the sample. Each of the plates had 10 microliters from the corresponding centrifuge tube, along with 250 microliters of the host bacterium and 3 millimeters of phage agar. The non-agar plates were placed in an incubator for 24 hours. The results indicated that 10 to the 1 serial dilutions plates indicated high concentration of plaques considered as a wet plate. This plate was further collected to generate concentration of phage solution known as a lysate. This lysate was collected by flowing it with phage buffer, which allowed this fusion between the two. The solution is then harvested by filtering it through a 0.22 microliter filter to remove agar particles and bacteria. This lysate was used for many basic experiments such as a spot titer and full plate titer. So going on from that, the spot titer was completed to determine the concentration of phage particles in the lysate solution. This protocol was completed by performing 8 serial lysate dilutions, followed by a spot test for each dilution, and then counting the number of plaques from one agar plate. By taking into account the volume and dilution used to generate those plaques, we can calculate the concentration which is present at the number of plaque forming units in every millimeter of lysate. Using the formula right here, the titer from spot 10 to the 6 was calculated and gave us 6.0 times 10 to the 9th. And then going from that, the next step was to calculate the titer from 10 to the 6th to perform a full plate titer on 10 to the 5th and 10 to the 7th plate to determine the concentration of phage particles in the lysate by using a plaque assay as mentioned before. These dilutions were plated because they were predicted to yield a countable number of plaques from the calculation in the previous spot titer. Thus, 8 dilutions of the spot titer protocol were performed, and 10 microliters were pipetted from each dilution into 250 microliter host bacterium into 3 different agar plates. Positive results were yield of a countable number uh, of plaques. As you can see over here, approximately 30 plaques on 10 to the 6th plate were counted, 1 plaque on 10 to the 7th plate, 6 plaques on 10 to the 6th, and 1,088 plaques on 10 to the 5th. And further calculations were able to make a wet plate from these plaques to obtain more phage lysate and continue the next steps of transmission electron microscopy, DNA extraction, as well as for long-term storage for future experiments. Phages, like most microorganisms, are too small to be seen with the naked eye. So to overcome this limitation, there is a transmission electron microscopy that uses a beam light of electrons instead of light. This allows magnification up to 10 million x. So when a phage sample is applied to a grid, the phage particles adhere to the carbon coating. So a lot can be learned from a phage based on its morphology. And phage physis was determined to have a cipher morphology with an 150 nanometer tail and 50 nanometer head. So going from that, using the high titer lysate from the full wet plates, we were able to character physis by analyzing its genome through DNA extractions. The importance of DNA extraction is because the lysate contains not only the clonal phage population, but also remains of lysobacteria cells, including bacterial nucleic acids such as DNA, RNA, proteins, and lipids. The presence of bacterial DNA and RNA, as well as protein and lipids, will interfere with future experiments such as electrophoresis. DNA extraction destroy the bacterial nucleic acid by using restriction enzymes. Physis DNA was extracted and digested with SAC2, NSP1, SALT1, HAY3, and HINDI3 restriction enzymes. The results were viewed with geoelectrophoresis So aside from HINDI3, all restriction enzymes were used to cut the DNA. Physis was characterized with restriction enzymes HAY3. Using data software such as Pecan, Galaxy, Famirator, PhageDB, BLAST, and next generation sequencing reveal that physis belong to cluster EG. The genome of physis has 62,078 base pairs, which transcribes rightwards 48.1% of the time and leftwards 51.9% of the time. Physis GC content is 68.2%, and it has 103 predicted protein coding genes. Genes including instructional proteins, histine trinucleotide binding protein, hydrolase, and among others. So in conclusion, the genome of physis contains 103 predicted coding genes. Physis is also a cluster EG bacteriophage with a genome length of 62,078 base pairs, and its morphology is Ciphervira with an icosahedral capsid. And our next steps for physis will be to further analyze the protein coding genes and submit an annotation to GenBank. And support of this research was provided by Howard Hughes Medical Institute, CPhages, Tarleton State University, College of Science and Technology, and the Department of Biological Sciences. And I am happy to answer any questions. Thank you so much.
I guess I'll go first again. So, this is the third presentation we've seen on this. So what's the bigger picture that we're going for here when we're annotating these genes and stuff? You did mention some, but specifically for yours, where do you think it's going? Where do you think it could potentially lead to?
Well, as of going from annotation, we were able to discover different proteins and different genes. And going from that, we could also do future experiments. I know that in Tarleton, they also do seed genes to discover to further analyze other bacteriophages and identify the proteins and their functions.
Does yours belong to a group? Have they already kind of started classifying these into certain groups, and those certain groups have certain characteristics that they see? I know you said they're fissile genes, but so what do you know, how is that different from the last presentation to yours?
So it's different based on like different types of genes and how they compare to other bacteriophages. So using Famirator, PhagesDB, and other softwares, we were able to also like compare and contrast other phages and determine which one was closer to a bacteriophage based on their genes and proteins. So pretty much like each bacteriophage is it's different based on its proteins and genes, if that makes sense.
Okay.
Okay, I have a quick question. So I know there's one slide about to use the restriction enzymes to the DNA of the phages. So what's the purpose to use this restriction enzymes?
So the purpose of the restriction enzymes was, like I said before, it contains not only the clonal population, but it also contains the remains of bacterial cells, and extra proteins from the previous experiments. So what we want is just pure DNA from the bacteriophage, and that's why we use restriction enzymes to just clearly get just the genome of the bacteriophage and not other extra proteins or extra stuff that is not needed, if that makes sense.
Well, the restriction enzymes, well, for my lab, we always use this to cut the DNA fragment from maybe one vector, one DNA sequence, and we use this sequence this piece to do some further cloning or something else. So that's what I thought normally the restriction enzyme is doing. So so if you say the restriction enzyme is used to isolate the bacteria, is that?
Well, that's like, I guess that's one part of it, but
Uh-huh.
you said it could also be to cut the DNA.
Yeah.
So, but I just kind of just went off like based on like what I presented, but yes, I will look more into it.
Okay, thank you.
Thank you, Gustavo, for a great presentation. Our next presentation will be by Emily from the University of Mary Hardin. Again, judges, do you need any more time for anything?
I'm ready to go.
Okay, well then, Emily, if you're ready, you can share your screen and start the presentation.
Okay, can everybody see this? Perfect. All right. Hi, my name is Emily Martinez, and I am a senior at the University of Mary Hardin Baylor. And today I'm going to be discussing my research on barcoding algae from the Chlorococcum genus. To begin, I'm going to give y'all a little bit of a background on barcoding. Barcoding is a worldwide endeavor that began in 2003 with Paul Hebert and his team. They did a variety of studies ranging from domains to species just to test to see how specific barcoding can get and if it can really work. Their last study was a group of 200 species of lepidopterians. And they were able to find that the mitochondrial gene cytochrome C oxidase 1 was able to differentiate between each of the different species of the lepidopterians. Now, this is important to my study because it was able to show that if you do find the correct gene, you can
tell the difference between each of the species based on genetics alone as opposed to physical aspects, which tend to fail us. Now, barcoding is what you do whenever you use a short sequence of DNA from a specific gene to be able to identify it as opposed to the physical aspects. Now, the importance of barcoding, as I've previously stated, is the identification of species. As you can see from the pictures I have up on the slide, even if they're not within the same genus, the species can still look extremely similar and can be mistaken for each other. So it would be more beneficial to be able to barcode it and look at the genetics of it, as opposed to just the physical aspects, because they could be mistaken for one another and not be identified properly. Another reason that barcoding is important is because it can be uploaded into a database. So if barcoding is more widespread and more research is done with it, then more species are able to be barcoded. And if someone who had an unknown sample was trying to figure out what their sample was, they would be able to upload the sequence of DNA that they were able to extract and run it through something like BLAST and figure out what their sample is based on previous barcoding. Now, some reasons for barcoding algae are algae is the source of half of the world's oxygen. Also, a lot of ecosystems are dependent on it. So bigger animals like humans or lions or something like that are dependent on smaller animals that use algae as their main food source. So naturally, I think that we would want to know just a little more about this little organism that provides so much and sustains so much life. The normal identifiers for algae are normally the way that we identify the different species. So we have shape, color, and size because it varies throughout all of algae. We have the spiral shape, we have these little horseshoe shapes, we have rectangular shapes, and we have many different colors of green, red, yellow, brown, and other colors. Not to mention they all come in different sizes. However, within similar genuses, we have a lot of species that just look extremely similar, as we can tell from the Chlorococcum genus and the Chlorococcae family. So as you can see, I have 3 of the species up on the slide that I was able to work with, and I have Diplobionicum, Scabellum, and Acidum. So as you can see from these pictures, all of these have the similar shape of them all being small, round. You can also see from the Diplobionicum and the Scabellum, they have a very, very similar tint of green to them, as well as them both being around the same size. So they're not able to be identified based on the normal identifiers of size, shape, and color because they all look extremely similar. There are also 38 different species within this genus, and I was only able to work with 10 of them, but they all look extremely similar to all of these that I have depicted, as well as they all grow very similarly. So it's hard to differentiate based on looks alone. So we would need genetics to be able to identify them. I was able to work with 10 of the Chlorococcum species, and these are the ones that I was able to work with. I was able to work with Scabellum, Diplobionicum, Aquaticum, Oleophasians, Sphacosum, Tetrenzi, Citriform, Macrostigmatum, Microstigmatum, and Lacustri. Now, the methods that I used throughout this research was I began with DNA extraction, or at least attempting at it, with the Biorad nucleic acid extraction module and the Zymo Quick DNA plant seed mini prep kit. Now, I say attempt because I was only able to get DNA with a good concentration and a good purity from the Zymo Quick DNA plant seed mini prep kit one time. And that was the very first time I was able to do it. And every time after that, even with changing my methods and using different beads for the Zymo Quick DNA Kit and trying different things, I was only able to get about from 1 to 5 nanograms per microliter for my DNA concentration, and they weren't very pure. However, I was running out of time and I needed to see if I could actually use it for PCR. So I went ahead and just used my not very pure, not very concentrated DNA and just ran it on PCR to see if it would work. And it did. And I got good results on my gel. So I ended up sending that off for sequencing and was able to run some bioinformatics on it using the CAP3 sequence assembly program and the Clustal Omega program. My results were I was able to use 2 different genes throughout my research, the TUF-A gene and the ribulose bisphosphate carboxylase large subunit gene. My TUF-A gene, I only used 2 of my species on because that was the first round of DNA that I did. And those 2, the oleophasians and aquaticum, were what I got a good concentration and good purity on. And then I was able to run the ribulose bisphosphate on 8 different species because other than the aquaticum and the oleophasians, those were the ones that I didn't get very good purity or concentration on, yet it was able to be run through PCR successfully. So these are my results from aligning them in the Clustal Omega program. And as you can see from the TUF-A primer, this is a decent barcoding gene because this one has over 30 different spaces where the nucleotides differ from each other. And that's what we're looking for in barcoding. We want to be able to tell the difference between them based on one gene. Now I use 2 different ribosome large subunit genes for these. And as you can see from this, it does have a decent amount of differences between them. However, it's not as good as the TUF-A gene because the TUF-A gene had more differences from it. So if I was looking for a good barcoding gene, I would use the TUF-A gene as opposed to this one. Now, in the future, I would like to look at more species under the TUF-A gene to see if it would be able to differentiate between more and not just the 2. So I would like to expand it in that. I would also like to use different reverse primers for the ribulose gene because my reverse primers that I did with the ones that I was able to show on the previous slide were not good with the sequencing. The sequences came out not great. And so I think that pairing them with different reverse primers would be able to show a little bit more variation, hopefully. I would also like to use or try the MAT-K gene, the Maturase K gene, as a barcoding gene because it has been shown with different species to be good for barcoding and have high variability between species. So I think that it would be worth it to look into that for the Chlorococcum genus. This is my bibliography for some of my background information. And I would just like to take a moment to thank Dr. Kathleen Wood for being my advisor and helping me through this process. CL and Gertrude York Professorship in Biology for the funding for this project, and the University of Mary Hardin Baylor for allowing students to partake in research on campus. And now I'll take any questions you'll have.
Okay, so I think I will go first this time. So do you have some like the gel images from your DNA purification or the PCR, like the PCR confirmation from the gel images?
I do, but I do not have them on my laptop here. They are actually at on the computer at the university.
Okay, so because I think if you have some more pictures about the gel images, that would be helpful.
For sure, I'll keep that in mind. Thank you.
Just a comment. Yeah.
Dr. Harville, I think you're muted if you're trying to ask a question.
Yes, I'm trying to ask a question. I'm just mimicking here stuff. How would we use this in, what would this be important to be able to tell the differences between two species of algae, and how fast and how costly would it be to do this as far as the barcoding stuff?
As far as barcoding, so as for how quick it could be, once we figure out a gene for it, it could be pretty quick. All you really need to do is run the PCR with your DNA, or I guess extract DNA first and then run the PCR with it, and then send it off for sequencing. So the PCR and the DNA extraction could be done in a couple days, depending on how much time you had to spend on it. And then however long wherever you send it off for sequencing would take. So that range, I guess, would vary based on who you use to sequence. As for how costly, I am not completely sure. My professor was the one that was buying all of the primers and stuff, so I'm not positive as to that, but I can look that up for you.
So where did you get your original strand, so that when you had your algae, you're like, okay, it's 100% species A and I'm doing this. So where did you get those original strands?
For sure. My professor, actually, Dr. Wood, ordered my strands from UTEX. And so we were able to actually know what it was, so that we're not kind of going in blind trying to figure out what it is while trying to barcode it.
And I guess my last question is, because of, just constant environmental change and adaptation and stuff, I mean, could it be that you sort of barcode it, and then maybe certain environmental factors that we're, causing, that they could change their code and therefore we can't identify it? Is that a possibility, or?
I do believe that would be a possibility. I think that it would be maybe less likely if we were to choose a gene that didn't have as much of a chance at mutating or becoming highly variable. I also note that it's not an RNA gene, and so it's not as likely to mutate as a DNA gene would, because of the DNA that is able to proofread.
Would that, I'm thinking now, just kind of thinking ahead, if we got it all barcoded, then we could actually monitor change.
For sure, yeah.
So it would actually be good, but it's just so timely, costly, and there's just so many. So I just think there's just some really cool applications here. Thank you.
For sure. Thank you.
Thank you to all the presenters who did a wonderful job. This concludes the Biology 3 division of HOT.